today is the last class for you for this semester. You know that, right?
Yes, sir.
Okay. Okay, so the basic characteristic properties of DNA molecule includes, so you see that there are different aspects which we have discussed about. There are enzymes which are required for the DNA synthesis, right? And the requirements are like DNA enzymes and all. But besides that, you should also see in what direction the replication will start, will take place. So let's imagine this is the template strand. So in this figure, this one is the template strand. So when it is like 3' to 5' end, and then you can see always the other strand will synthesize only in the direction of 3' to 5'. So that's what you see here. The synthesis is always in the 5' to 3' direction only. So that means you see when it is having 3', that means the template strand, and then the 5' is the beginning. So then it will continue like this, right? So if there is another strand, let's imagine there is another strand here that is like 5' to 3', right? So that means up to here. Then how the DNA synthesis will begin? So the DNA synthesis will begin always in the 3' end of the template, and then it will continue towards the 5' end of the template molecule. So that means the newly synthesized strand always starts with the 5' end, and then it ends with the 3' end. So the direction of replication process is always in the 5' to 3' direction. And then you can see how the nucleotide or the nucleotides will bond one to other. Then you see this is 3' end and this is 5' end. That means this is the beginning here. So the new strand is getting synthesized. All the nucleotides will get joined one to other, and then they form as a phosphodiester bond, this yellow one, right? So the phosphate with the 4 oxygen molecules, we call it as phosphodiester bond. So the phosphodiester bond will form, and this phosphodiester bond always helps to find what we can say the bonding between the one nucleotide to the other nucleotide. So that's the one which makes the connection between the nucleotides. So the nucleotide is having a nitrogen base and then a sugar. In case of DNA molecule, it is a deoxyribose sugar, and then the phosphate will be there. So these three together can be called as nucleotide, and one nucleotide will join to the other nucleotide through the 3' OH group of the ribose sugar, that's this one, and then the 5' phosphate group of the upcoming nucleotide. So that's why you can see this is upcoming nucleotide and this is already there, and the 3' OH group of this deoxyribose sugar will be acting with the 5' phosphate group, especially the alpha phosphate will involve in the formation of phosphodiester bond. So because if you talk about nucleotides, nucleotides are like they are in the triphosphate form. So let's imagine ATP. ATP is a adenosine triphosphate. So the adenosine in the sense, so it has adenine group, nitrogen base, and then ribose sugar, deoxyribose sugar, and then triphosphate. You can call it as like adenosine triphosphate or deoxyadenosine triphosphate, something like that. So it may be DNA or RNA, whatever it is, whatever nucleotide it is. Okay, so that's what we can see. So anyhow, the upcoming nucleotide is always having triphosphate at the 5' carbon. So the 5' end or the 5th carbon is holding that, and out of all these three, only the alpha carbon, that means the alpha phosphate, that is immediately at after the carbon atom, 5' carbon atom. So we can call it as like alpha carbon, sorry, the alpha phosphate. So this alpha phosphate can interact with the 3' OH group of the deoxyribose sugar, and then it forms as a phosphodiester bond. And this is having like the pyrophosphate, right? Two phosphates, and then PO, it's P2 P2O5 minus, P2O5 minus will be there, right? This is what we can see. So here what will happen, so you can see these two phosphate molecules will be released in the form of pyrophosphate, and the what we can say, like, the phosphate will join to this hydroxyl group, and so that it forms as what we can call it as, it forms phosphodiester bond. So that phosphodiester bond is always helps to find the to join one nucleotide to the other nucleotide. Okay, so once the nucleotides are joined, then what will happen? Because these nucleotides are always joined based on the sequence which is present in the complementary sequence, or you can also say like template strand. This is the template strand. You see the template strand. So when the template strand is there, and nucleotides are there in the same template strand, so it is having guanosine. We know that the complementary nucleotide for the guanosine is always cytosine. So guanosine always pairs with cytosine, and then adenine always pairs with or thymine. So it's vice versa. When we have a guanosine, so the guanosine definitely it should accept only the cytosine molecule to be having a complementarity. So it forms as 3 hydrogen bonds in between these two nitrogen bases, and whereas like adenine and thymine, in between them we can see there are 2 hydrogen bondings. So likewise, the upcoming nucleotide, so which nucleotide has to come as an upcoming nucleotide is decided by the sequence of the template strand. So the template strand, you see here, the template strand is having nucleotide sequence, you see, continuously, and this nucleotide sequence will decide which nucleotide should be added to the next in the polymerization reaction. So that you can see. So now in the same way you see here, so the same way, see for example, usually the polymerase, DNA polymerase is the enzyme which is involved in synthesizing the DNA molecule. But this DNA polymerase usually it continues, but it doesn't have the capacity to synthesize the primer molecule. It doesn't initiate the replication process. So for replication process, I already told you in the previous classes, a primase is an enzyme which can initiate or which can, what I can say, like, which synthesizes the RNA nucleotides. So other RNA nucleotides can be called as a primer nucleotides. You see, beginning of the DNA synthesis or the DNA synthesis initiation is done by the primase enzyme. So the primase enzyme always continues, so that it synthesizes more, like, the 9 to 12 nucleotide sequence. Only 9 to 12 nucleotide primer molecule can be synthesized, and afterwards you can see the 5 prime to 3 prime. You see, so because it is already having a 3 prime end, I don't see this. The 3 prime end, a 3 prime end is always providing a OH group, 3 prime OH group, and that's why the DNA polymerase is the enzyme which polymerizes the 5 prime phosphate with the 3 prime OH group. That's the only thing it has done. Okay, so that means it doesn't have any other function that it can add a nucleotide without any 3 prime OH group. It doesn't have any character, such a character. So it adds only when there is a template strand available. So that means this is the template strand, right? This is the template strand, and then 3 prime OH group is available, and then the DNA polymerase will recognize this, and then it continues to synthesize, add the nucleotides, which are complementary to this nucleotides. Okay, template nucleotides. That's what we can see with the DNA synthesis. Okay, so anyhow, the DNA polymerase, sometimes it make mistakes. You see here, the DNA polymerase is added a nucleotide. You see, we know this green always, pairs with the pink, but you see that the pink is added to this, and yellow is always with the blue. You see, do you understand this? Is it clear, ma? I want answer from any one of you. Is it clear?
Yes, sir.
Is it okay? Okay, so what will happen here? So because a wrong base is incorporated in this case, when the wrong base is incorporated, you see the polymerase adds an incorrect nucleotide. So the wrong base is incorporated here. When the wrong base is incorporated, then immediately the DNA polymerase is having a capacity that it has a function, what we call it as like 3 prime to 5 prime exonuclease activity. 3 prime to 5 prime exonuclease activity. So the nuclease nucleases are the enzymes which are present in the nucleus, and nuclease activity always tells you that it cleaves the DNA molecule, or it degrades the DNA molecule or RNA molecule or any nucleic acid. So the nucleic acid will get degraded. So when the nucleic acid is degraded, then automatically what happens? What will happen? A nucleic acid, when it degrades, then you can see that the nucleic acid always, it leads to the degradation of DNA molecule. When the DNA molecule is degraded, the cell will die, right? So that is how the nucleases will work inside the cell. But the DNA polymerase enzyme is having exonuclease activity. So there are two kinds of nuclease activities which we can observe. One is endonuclease activity, and the other one is exonuclease activity. So the exonuclease activity always starts with the ends. So that means it removes the nucleotides from the ends. So you see that this is the DNA molecule, and usually the exonuclease activity or the endonuclease activity will work only when it is in the single stranded form. Okay, double stranded forms may not be get affected, but endonuclease activity can be shown even in the double stranded form, so it can cleave the DNA molecule. Okay, so that means endonuclease in the sense it cleaves in the middle of the DNA molecule anywhere, and exo in the sense it start removing the nucleotides from this side or this side. That means any one of the end. So it recognizes and then it removes all the nucleotides. So here the DNA polymerase is having 3' to 5' exonuclease activity. What do you mean by the 3' to 5' exonuclease activity? So that means it has the capacity that it can remove the nucleotides from 3' to 5'. So in the growing polynucleotide chain, so there is a 3' is always in the beginning. So that means where the DNA polymerase is also attached. So when it is attached, automatically what will happen when there is a wrong base incorporation? So the unit or subunit, a protein which is responsible for the 3' to 5' exonuclease activity in the DNA polymerase complex will recognize this, and then it removes some of the nucleotides. Like 2-3 nucleotides will be removed back. So then again the DNA polymerase can continue. That means it will add the right nucleotide. So this kind of function of a DNA polymerase is called as 3' to 5' exonuclease activity with proofreading activity. So the function is called as proofreading function. Are you clear? So that means it has the capacity that if there is any wrong base is incorporated, then automatically it goes back and it check the back nucleotides, and then it will say that it removes some of the nucleotides, and then finally what will happen? It corrects the nucleotides. So it adds the right nucleotide into the chain. So that's why the fidelity of the DNA polymerase is very high. A fidelity in the sense accuracy of the DNA polymerase in its action is 100%, almost 100%. So you see that in case of E. coli, the number of mutations will happen for in the genome is like for 1,000 cycles. You can see that one mutation will take place. For 1,000 cycles, for 1,000 replications, see that only one mutation will take place because of the proofreading activity of this DNA polymerase. This is what we will see. Are you clear? Is it clear? So the mispaired nucleotide will be removed, and then 3' to 5' exonuclease activity or proofreading activity of the DNA polymerase can correct it. You see, so it is corrected. It's removed, and then it will continue. It go back, then it will come back again. It moves. You see the new the correct nucleotide was added. This right. This is what happens with the DNA polymerase, right? So do you see this slide? Emma, is it visible?
Yes, sir. Slide number 9.
Yes, sir.
No, is it visible now?
Yes, sir.
So I changed the next one. I went to the next one. So it's like, why does DNA replication only occurs in 5' to 3' direction? Is it the same slide?
No, sir. Synthesis
No.
is always in the 5' to 3' direction.
No, that one is the previous one, right?
Yes, sir.
Now Can see? Is it visible or not?
సార్ చేంజ్ అవ్వలేదు.
అవ్వట్లేదు. Okay, so now you can see, right?
Yes, sir. Yes, sir. It's visible, sir.
Yes. Okay. Okay, so why does the DNA replication will takes up only 5 prime to 3 prime direction? So what will happen here? So when we talk about the 5 prime to 3 prime direction, so the hypothetical reaction here, I already told you in the previous slide. So why it is happened? Because the DNA polymerase always requires a specific kind of reactivity. It can join the two molecules by forming the phosphodiester bond when it has a 3' OH group in the ribose sugar, deoxyribose sugar, and then what will happen? And then the 5' phosphate group. And the phosphate is always having, like, it should be what? It should be always having triphosphate. Okay, it should be always having triphosphate. Then only it can be acted as a, it can be acted as, what we can say, it can join the 3' OH group with the 5' phosphate group. If there is only single phosphate at the 5' phosphate, 5' group, and then it won't be joined. So there is another enzyme which can be used for that, for joining. Okay, but remember, the DNA polymerase is the one which is having always responsible for, it's always responsible for joining the molecules. So the joining of the molecules is always, joining of the molecules is always done through 3' OH group. That means it should have like OH group and then one phosphate group, and the phosphate group should always be the alpha phosphate. So the nucleotide should have always triphosphate. Then only it can be happen. Okay. Okay, 1 second, ma. Okay, so then what will happen here? So the hypothetically, you see, this is 5' to 3', it is going up, and then you can see here the upcoming nucleotide is going to join here, you see. So the upcoming nucleotide will join, right? So then a proofreading activity will be done. So that means it's all like, proofreading and all will be done, and then 5' end produced if one nucleotide is removed. So that means usually initially what will happen, so it forms as like triphosphate, but later on what will happen, the triphosphate will get removed and then only the single phosphate will be joined, and this process I'll see in the later. Okay, so because I'll tell you this when we go for complete process of replication. Okay, how it happens and what will happen, and these things we will see later. So here what will happen if it is in the 5' to 5' direction. So this is like, A, figure A represents what will happen if the nucleotide is added at the 5' end. Let's imagine there is a 5' phosphate group and then 3' OH group, and then what will happen, the 5' will become into triphosphate. Because I already told you the DNA polymerase is having an activity that it can bind only to the 3' OH group, because when it has like a phosphate group and then the upcoming phosphate group upcoming nucleotide cannot be joined here, right? You see the next upcoming nucleotide. So because of the proofreading activity, what will happen, this will be removed because this is not the appropriate or the proper nucleotide which need to be added, right? So the next nucleotide will be added here. Reaction does not proceed because as no high energy bonding between would be cleaved. So there is no high energy bonding cleavage. High energy bonding cleavage in the sense, that the phosphate is the one, triphosphate is there, and the pyrophosphate will be removed. So here the high energy bonding should be cleaved. So that will happen. But whereas in this case, when it is in the 5' to 3' direction, and actually, so the nucleotide, upcoming nucleotide will be added here. So it's like, this is 3' to 5' strand, and this is 5' to 3' strand. A proofreading activity will done, so which leads to the addition of this 3' end produces when one nucleotide is removed by the proofreading, and then incoming proofreading activity. So that means incoming nucleotide will be joined to this. Then the pyrophosphate will be released, and then this nucleotide with the single phosphate will go and join to this. That's what we can say. That means high energy bond is cleaved, but whereas here there is no cleavage. You see, the pyrophosphate cleavage is the high energy bond cleavage, and which provides the energy for polymerization. So this is what we'll see with this molecule, right? You see why the DNA molecule always, or the DNA polymerase can polymerize the DNA molecule only in the 5' to 3' direction. So why it is? It is because the high energy bondings, high energy breakage or high energy release will happen only in the 5' to 3' direction, but not in the 3' to 5' direction. So this you have to remember. Okay. So there are two kinds of replications which we can see here. Is everyone joined into the class? So only 26. So what is happening with the others? Okay. So let me check. Okay. So I already told you what is the like no direction of DNA replication, because when I told you, so there is a primer molecule is in the DNA molecule. That means when the new strand starts. So when the new strand synthesis begins, then the RNA primer will be joined there, and then this RNA primer will always helps to find the, or you can say that helps to find the DNA polymerase to continue the DNA polymerization process. So there are two kinds of replications which we can see. One is called as unidirectional and the other one is called as bidirectional. I already told you about this, but you just see in the pictures where which one tell you what. So the unidirectional replication is always represents. So that means the replication will continue. So it begins at one place. You see, it begins at one place and then it will continue. So this is the new strand which is going to be synthesized. So the unwinding will take up likewise. So heavily labeled DNA molecule is this one, and then lightly labeled DNA molecule is the another one. So, but you can see here in both the strands what is happening. The DNA replication is started at one place and then it is moving further towards. You see, but it will not happen. You remember. So why it is not happen? Because the double stranded DNA molecule have a different kind of pattern, that it has like 5' to 3' end for the one strand, and whereas the other strand in the opposite manner. So that means if one strand is having 5' here and the other strand is having 3', right? So if it is the case, I already gave you one example in the previous slide, that DNA polymerization will take place always in the 5' to 3' direction. Remember, not only DNA polymerization, so the 5' to 3' direction is always meant for, like, the transcription process where the RNA molecule will get synthesized, and also you can see the protein translation can also happen on mRNA molecule by reading 5' to 3' direction only. So it's always 5' to 3' direction only. Okay, so the molecular biology experiments always says the 5' to 3' direction experiments, right? So in the unidirection, you can see that the DNA replication will takes up only in one direction. You see here in the first picture, unidirectional replication, where the DNA synthesis began here and then in one strand, and in the other strand is also in the same direction. You see, and new strands are getting synthesized. So here it is red, here it is blue. That means that the opposite strand, it seems, right? So what does it mean? Two reactions will happen, but this will not happen. This will not happen in the biological system. Remember, unidirectional replication will happen only in the plasmid molecules, only for up to certain stage of replication, and later on it will become into the bidirectional replication. Do you understand what I mean? Are you clear? Right? So the bidirectional replication will take up. So what will happen here? The unidirectional replication will not be happen like this. This is not the exact way, because it won't start in the same place for both the strands for the synthesis. You can say one direction, one nucleotide, the one strand is synthesizing in this direction and the other strand is synthesizing in this direction, then it's okay. Then we can call it as like a unidirectional again. So, but you can't see this kind of replication in the biological system, but you can see the so in the bidirectional replication again, you see one strand is started synthesizing the new strand in this way, and whereas the other strand in this way. So that means this is the beginning for this strand and this is the beginning for this strand. You see, right? So these are the two things which we can see. Whereas this is the just approaching or the polymerizing end. So again here the polymerizing end. So the two DNA molecules when they are, so one DNA strand will continue like this and the other strand will continue like this. So that we can call it as like bidirectional. So that means not in the same direction, they are in the different direction. So one is in this direction, one is in the other direction. So that we can see as a bidirectional replication. Okay. So the next one is semi-discontinuous replication, because we always tell that so we have like two kinds of replications. One is continuous replication and the other one is semi-discontinuous replication, right? Discontinuous or semi-discontinuous replication. Let's see what will happen here. So there are two anti-parallel strands which replicate simultaneously, which is leading to the leading strand synthesis. So the leading strand synthesis is always takes up in a continuous manner, whereas the lagging strand synthesis will takes up in the fragments manner, where like 5 prime to 3 prime direction the replication will takes up and then they will be joined later. Okay, let's see how it happens in the picture. So in this picture, this is the replication fork movement. So where we can see, and the double stranded DNA molecule is getting unwinded. So let's imagine that this is the strand which is having 3 prime to 5 prime end, the blue one, and the red one is having 5 prime to 3 prime end. I already told you DNA replication will happen only in the 5 prime to 3 prime direction in the newly synthesized strand. So the 5 prime to 3 prime direction in the sense, so it always in the opposite manner in case of DNA molecule. So the complementarity should be maintained. So that's why what will happen, the 3 prime strand which is having the unwinding portion will be involved in the continuous DNA strand synthesis, and that's why this strand can be called as, that means the strand which is getting synthesized is called as leading strand. That means it doesn't have any kind of breaks, so it continuously it is going. So because it is 5 prime to 3 prime and all the time the DNA molecule is getting unwinded, then that's why what will happen, the 5 prime to 3 prime direction will continue, right? So this is what we can see, this is what we will see. So the leading strand will always be continued, then automatically you can see that the leading strand will have a continuous synthesis of DNA, right? And whereas in the opposite strand, because I already told you always the DNA synthesis will start from 5 prime to 3 prime. So when this DNA molecule is having 5 prime, then automatically so it starts with the 3 prime only. That means the complementarity will always start ends with the 3 prime but not with the 5 prime. So if you want to start the 5 prime to 3 prime 5 prime to 3 prime synthesis, then automatically see in this case when the DNA molecule is unwinded up to here, so it recognizes that I already told you that primase is the enzyme, right, which recognizes the primer synthesis. It adds the 9 to 11 RNA nucleotides to the initiation step, right? So same way, because this complex of this protein molecule or the DNA polymerase is also having this which we call it as like replicase. Repliase is a complex enzyme which has like DNA polymerase and all other subunits, and including helicase, ligase, primase. So there are so many enzymes are present in the same complex. Okay, so the primase is present, and then primase will recognize a specific nucleotide sequence on the template strand, and so that it synthesizes the RNA nucleotides. So once it synthesizes the RNA nucleotides, then automatically it's always 5 prime to 3 prime. You see, this is 5 prime, but when it is going this direction, then here the 5 prime, and then it moves along, right? So when it provides the 3 prime OH group, and then automatically DNA polymerase, here it is, it can recognize and then it will continue. Okay, so up to here, so it will continue, and then next, then unwinding will takes up, and when it reaches here, then automatically again these nucleotides are recognized by the primase, and then it synthesizes the small stretch of RNA nucleotides, and which provides the 3' OH group for the DNA polymerase, and then it will continue. So that means it goes up to here, but it is already double stranded form here. So that's why it will continue and it will synthesize up to here. And this is RNA nucleotide, and then RNA nucleotide in the sense it has like 5' phosphate group and then a 3' OH group, but even then the DNA polymerase cannot add these nucleotides. Okay, so what is happening here, because it is having RNA nucleotides here in the primer, this primer is having RNA nucleotides, and that RNA nucleotides cannot stand in the DNA molecule, so they have to be removed. Okay, and then what will happen, the DNA polymerase 1 is another enzyme, because until now we saw that the DNA polymerase is like DNA polymerase 3. Okay, there are three kinds of DNA polymerase 1, 2 and 3 in case of prokaryotic system, whereas in eukaryotic system there are so many that we'll see later. But anyhow, the DNA polymerase 1, 2 and 3 are the three different types of what we can call it as like nucleotides or the enzymes which are responsible for the DNA synthesis. So the major DNA synthesis, that is polymerization step, is led by the DNA polymerase 3, whereas a DNA polymerase 1 is the one which is responsible for synthesizing the, or you can say like repairing the DNA molecule. So how it repairs? So when this DNA polymerase 3 is continuing here, then DNA polymerase 1 will recognize this, and then it removes the RNA nucleotides here. When it removes the RNA nucleotides, then automatically, because here it is 3' OH group and there is complementary nucleotides, then automatically the DNA polymerase 1 will continue to synthesize up to here. But there is a gap. What will happen here? The gap is like, it is having a phosphate and 3' OH group. And the DNA polymerase doesn't have any capacity that it can add the 3' OH group with a single phosphate group. It requires triphosphate, only alpha phosphate will be joined to that. So that's why what will happen is it won't be join this nucleotides. So then a ligase is an enzyme. So ligase enzyme is basically, it can add the nucleotides based on, so in between the single phosphate and the OH groups. So that's why that enzyme simply the 3' OH group is available here, and then 5' phosphate group is available here. So then automatically they will join together. So when they join together, they form as phosphodiester bond. That I'll tell you in later classes. Okay. So how the DNA ligase can act and all that we will see. Okay. So what are the different factors influences this and all that we will see in the later classes. So finally what will happen? So this fragment will join with this, and then similarly this fragment will also join with this by the DNA fragment, DNA polymerase 1. So likewise what will happen? So here you can see it's not leading, that means it's not continuous synthesis. That's why it can be called as lagging strand synthesis. And in the lagging strand synthesis, the fragments were generated maybe something like they were like 200 to 2000 nucleotides in length. So they will be called as Okazaki fragments because the scientist who discovered this is Okazaki. So that's why after him the fragments are named as Okazaki fragments. So these Okazaki fragments are later joined together by the action of DNA polymerase 1 and ligase, DNA ligase, and so that they will join together. So when you talk about see, when it is having break, and then how come they can be joined? So they will be joined by the action of DNA polymerase 1 and DNA ligase. So this will happen, this we will see. Right? So similarly what will happen? So the other strand, leading strand, always continue to synthesize, there is no break, and whereas lagging strand is always synthesizing a small stitch of RNA, and then DNA nucleotides which may be like 200 to 2000 base pairs in length. So they will be joined later together by the activity of DNA polymerase 1 and then a ligase. Okay, DNA ligase, so that they will join together, and then finally at the end what will happen? So we can see that all the two strands are continuously synthesized. That's what we can feel. But you see that one strand is always involved in the lagging strand synthesis, and the other one is involved in the leading strand synthesis. Right? So we can say the continuous direction is always like 5' to 3' direction when the template is available, and then the fragments When the template is not available, then what happens? The fragment will be generated. So where we can see they can be called as Okazaki fragments. That's what we can see. Right? Then the next is semi-discontinuous synthesis, semi-discontinuous replication process. How it happens? So the new strand synthesis always in the 5' to 3' direction, which we already know. So semi-discontinuous, even in the previous slide, I already told you, right? So in the semi-discontinuous process, see the new strand will get synthesized. You see, this is 3' to 5', the template strand. So that's why the replication will begin from 5' to 3'. So the continuous strand synthesis will happen in this direction. So the upcoming nucleotide will come, and then finally what will happen? The two strands will form. Whereas in case of the other strand, so this is called as like leading strand and this is a lagging strand. When it has, you can see, you see this is 5' and this is 3'. So there is no way that the 3' to 5' uh, polymerization activity is lacking in the DNA polymerase. So that's why what happens, as and when there is a special sequence and recognized by the DNA polymerase, the primase enzyme, and then it synthesizes the RNA, stretch of RNA nucleotides. So ribonucleotides, and these ribonucleotides will provide 3' OH group, and this 3' OH group is recognized by the DNA polymerase 3, and then it will continue to synthesize until the end of the uh, gene, or like until the opening of the gene, you can say, right? And similarly, you can also see that the same kind of sequence when we see, so there again what will happen, the another strand will start synthesizing, so that likewise there are so many fragments will be generated, which has been shown here. You see, they can be called as like lagging strand with Okazaki fragments are present here, and then most recently synthesized DNA molecules are near the replication fork. So they are always near the replication fork. That's what we can see. Is it clear? Is this clear? అర్థమవుతుందమ్మా?
Yes, sir.
Sure, everyone?
Yes.
Yes, sir.
Okay, great. Okay, so then how RNA primers will form? You see here, in the DNA replication process, the beginning, what will happen? The RNA primer will join here, and then so this may be replaced, or not now, maybe later on it can be get replaced. But anyhow, here what will happen? The 5' to 3' direction will be happen in a continuous manner, but in case of Okazaki fragment formation, you can see RNA primers are formed based on a specific kind of nucleotides. So that specific nucleotides are recognized by the primase enzyme, and then it will continue to synthesize the RNA primer molecule, right? So this is what we can see, and you see in the next picture. In the next picture, you see the first strand is already synthesized Okazaki fragment, and second one is started here. That means as and when the DNA molecule is unwinding, then automatically what happens? The primer synthesizing nucleotide sequence will be identified by the primase enzyme, and then it adds the RNA nucleotides. And so when there is a gap, template, and then 3' OH group, and then DNA polymerase will continue to synthesize that. That's what we can see here. Is this clear? Right?
Yes,
Okay,
sir.
so what are the different types of proteins that are present at the replication fork? So how we can see them? So there are so many types of enzymes. I already told you the different types of enzymes are involved in the DNA replication process. So the first are like, topoisomerases. Topoisomerases are the ones, the enzymes which are responsible for releasing the torsions by the DNA. During the replication process, what happens? The unwinding happens. So because of the unwinding, at the end of the DNA molecule, you can see there is torsion generated. That's what I already told you. Take a thread and then uh, knot it at the one end, and then hold it on under your leg at one end, and then uh, pull the two strands from the top, then you can see that there are some knots are formed, right? So in case of DNA molecule, when that knots are formed, so the DNA is very sensitive, so that's why it may be broken. So that breakage cannot be happen because of the topoisomerases. Topoisomerases will cleave the DNA molecule and then they release the torsions. They release the torsion. And next is a helicase enzyme. You know the importance of helicase. Helicase enzyme always when the double stranded DNA molecule is there, so it synthesizes like this. So that means it will, what we can call it as, the double stranded DNA molecule will become into single stranded DNA molecule, so that it forms as a what we can say replication bubble. So it separates the double stranded DNA molecule into single stranded DNA molecule. Then the primase is another enzyme, and primase is responsible for synthesizing RNA primers. So it recognizes specific nucleotides, and those specific nucleotides, using them it synthesizes 9 to 11 RNA nucleotides, which will act as, which provides the 3' OH group for the continuation of replication by the DNA polymerase. Okay, so that's what we can see. And then the single stranded binding proteins. A single stranded binding proteins, because the helicase is the enzyme which unwinds the DNA molecule, right? So that means it converts the double stranded DNA molecule into single stranded DNA molecule. So when it is in the single stranded form, I already told you, endonucleases or exonucleases are very prompt to act against the single stranded forms, right? When it is in the single stranded form, then automatically the cells will have this endonucleases and exonucleases. These enzymes may cleave this DNA molecule, the single stranded DNA molecule. That's why in order to prevent the binding, or in order to prevent the cleavage of the single stranded DNA molecule in the nucleus by the nucleases, so the single stranded binding proteins will go and bind to it. So when they bind to it, and then automatically they won't allow the what we can call it as, like, DNA endonucleases or exonucleases, so that they may not recognize it, and then finally they can be, they can what we can say, like uh, prevents the binding of this nucleases to the DNA molecule, single stranded DNA molecule, such a way that it can protect the single stranded binding proteins, can protect the DNA molecule for the DNA replication, or like transcription, or whatever it is. Okay, next is of course the single stranded binding proteins prevent the re-annealing of single strands, so that's what we can see. And then they will also protect the DNA molecule from the nuclease activity. Then DNA polymerases are the important enzymes which we can see. They are involved in the synthesis of new strands. They are very specific and responsible for synthesis of new strands. How they synthesize the new strands? So by the polymerization process, they recognize the 3' OH group, and then they will add the another nucleotide, so that likewise what happens, it can continue to synthesize the DNA nucleotides, or polymerize the DNA nucleotides. Then the next is tethering proteins. So the tethering proteins are the proteins which are responsible to stabilize the DNA polymerases. So the DNA polymerases may be having a loose interaction with the DNA molecule during the replication process, that can be strengthened by the tethering proteins. So these proteins will help. Next is DNA ligase. DNA ligase is an enzyme which seals the nicks via phosphodiester linkage. But I already told you what is the importance of DNA ligase. DNA ligase is the enzyme which can DNA ligase is the enzyme which can bind to the DNA molecule when there is a sealing, because I told you the DNA ligase is responsible for sealing of DNA molecule when it has like a single phosphate group and the 3' OH group. So that means single phosphate and then 3' OH group will be joined by the action of DNA ligase. Whereas DNA polymerase can ligate the DNA molecule or can polymerize the DNA molecule when it has a 3' OH group and then 5' triphosphate group, right? That's why the DNA polymerase 1 cannot seal the DNA molecule. So the best example here is like replacing the RNA primer and then synthesizing the DNA nucleotides, and then joining of those nucleotides gap by the DNA ligase can be done. So the DNA ligase is like two types of DNA ligases we can see. One is like NAD dependent DNA ligase and then ATP dependent DNA ligase, NAD and ATP. So these things we will also see, right? Then the next one. Okay, so what is the mechanism of DNA replication? So in the yesterday class, I told you what is the mechanism. So the mechanism of DNA replication always. So it is invented by, like, the Korenberg, and then he got the Nobel Prize. Okay, so for the invention of the steps involved in the replication process. So basically he said that the DNA replication process is of having three major steps. One is called as initiation, elongation and termination. So these three steps we can see. So in the initiation step, what will happen? The proteins bind to DNA and open up double helical structure and prepare DNA for complementary phase, base pairing. So what will happen? Initiation. So there are different types of proteins. We can call them as like initiation proteins can bind to the DNA molecule, including the DNA polymerase or RNA polymerase, primase, replicase, or so many things. So all these will get attached to that, and then they will prepare for the complementary base pairing. So that's what we can see in the initiation. Elongation, proteins connect the correct sequences of nucleotides into a continuous new strand of the DNA molecule, into a continuous new strand of DNA molecule. And the termination will happen. So proteins release the replication complex, so that automatically what will happen? The replication will be ended, so the strands will get separated, right? So that's what we can see here. Okay, so when we see the mechanism of DNA replication, okay, so how the mechanism of DNA replication will take some. Do you see this slide? 4 proteins at the replication fork. This one.
Yes, sir.
Yeah, now you can see. Okay, so in the replication fork, there are different types of molecules which can be attached here. You can see, like DNA polymerase on lagging strand, and then you can also see that newly synthesized strand here you can see, and then leading strand you can see here. You see the leading strand is getting synthesized, and newly synthesized strand is here, right? So it is getting synthesized. And sliding clamp is the one, because I will tell you what are the different parts involved in, present in the DNA polymerase. And these parts are, you can say, I told you, like, it is having like 12 types of proteins of holding 16 proteins, right? So 12 types, but total is 16 proteins that we can see. Okay, so the sliding clamp and DNA polymerase on the leading strand. So one unit of DNA polymerase will be involved in the synthesis of leading strand. Other unit of the DNA polymerase is involved in the lagging strand synthesis. So that means you can see there is Okazaki fragments will be synthesized with the, because two units ఉంటాయి ఒక దానిలో. You see here, this one, it has 2 units. So 2 subunits, you can see all these, but you can see one part of the DNA polymerase is involved in the uh, continuous synthesis, and the other strand is involved in the Okazaki fragment synthesis. Okay, so here you see this is a single stranded binding proteins, so which can protect this DNA molecule from the degradation, right? So DNA helicase and DNA primase, or the primase enzyme is adding the primer nucleotides. Helicase is the one which unwinds the DNA molecule, double helical DNA molecule. And then topoisomerase is the one which cleaves the DNA molecule at one place, and then it releases all the torsions, right? That you can see. Okay, so what are the different types of enzymes which are involved in the DNA synthesis? So there are you can say, like, there are 5 major enzymes. I can say there are so many enzymes, but you can see the major enzymes are like 5 major enzymes which are responsible for the DNA synthesis. The first one is RNA polymerase. I'm not going to talk about the RNA polymerase here, but I will say the RNA, instead of RNA polymerase, I can say it's a primase. So the primase is the enzyme which is responsible to synthesize the 9 to 11 stretch of RNA nucleotides in the during the replication process. But we can call it as, because it is synthesizing the RNA nucleotide, so it can also be called as RNA polymerase. So that's why it is written as like RNA polymerase here, right? And then DNA polymerase is the another enzyme. We know that the DNA polymerase 3 is the major enzyme which is involved in the major replication part. So where the DNA polymerase is having a different kind of characteristic properties like 5' to 3' polymerization activity, 3' to 5' exonuclease activity, and so on. So there are so many activities we can see, and you can also see that the DNA polymerases are like different types, like DNA polymerase 1, 2, and 3 in case of prokaryotic system, and in eukaryotic system. So there is different types of DNA polymerases which you can say, like DNA polymerase alpha, DNA polymerase beta, DNA polymerase delta. Okay, so likewise there are so many DNA polymerases which can be identified. So anyhow, DNA polymerase is the major enzyme which is required for the DNA synthesis. Then the primase is an enzyme, so which is similar to the, or you can say like RNA polymerase. So this primase enzyme is always responsible for the primer synthesis. So the primer synthesis will happen, right? So it gets synthesizes. And next is helicase. So the helicase is the enzyme which unwinds the DNA molecule. So which unwinds the DNA molecule. So ఇలా ఉన్న DNA molecule ni, helical molecule ni, so it unwinds like this, right? That is how the helicase enzyme will happen, will be useful. Then the topoisomerase is the enzyme. Topoisomerase is always helps to find the knots which are generated due to the unwinding by the helicase enzyme, and so that knots will be released by cutting any one of the strand, or sometimes the two strands will get degraded, and then finally they will be joined together by the activity, right? So by the action. So as and when the topoisomerase cleaves, then automatically DNA ligase enzyme will go and ligate it. Okay, so this is what the enzymes we can see. Okay, so what will happen with the eukaryotic chromosomes then? Or they have multiple origins of replication? Is it true or false? Anybody can guess. You may guess. Juniors. You tell me. So this is the last slide which I am going to tell you today. So can you tell me, eukaryotic chromosome have multiple origins, or like only single origin? Single origin or multiple origin? Tell me. Emma,
It's
you
true.
just have a wild guess. I'll tell you what is it. Is it has? Do you understand what I mean? Whether it has a multiple origin of replication or a single origin of replication, eukaryotic chromosomes.
Multiples है.
Yeah, right. That is right. So because if it is only one, and then it takes very long time to synthesize the DNA in the eukaryotic system, but it is synthesizing in the synthetic phase itself, because there are multiple sites for the DNA origin of replication. So all the sites are responsible for the initiating the DNA synthesis, so that the DNA synthesis will begin and then continue, right? Is it clear? Okay, so if it is clear, and then I'm going to take your attendance. Do you have class at 11?
Yes, sir. Mansa madam.
Okay, great. Let me
Sir,
check
how is
your
your health?
Emma.
వ్యాక్సిన్ వేయించుకున్నారు కదా, నిన్న బాగాలేదన్నారు కదా, ఇప్పుడు బాగుందా అంటున్నాను సార్. ఇప్పుడు హెల్త్ బాగుందా అని అడిగాను సార్.
I'm all right. I'm all right. Even yesterday I'm all right. Only little uneasiness. That's it. Okay, so no worry. So I'm going to take your attendance and then I'll leave the class. Okay.
Sir,
చెప్తల్లి.
sir, good morning, sir.
గుడ్ మార్నింగ్ మా చెప్పమ్మా.
సార్ నిన్న క్వశ్చన్ పేపర్స్ తీసుకోవడానికి లోకల్ వాళ్ళు రమ్మని చెప్పినారు కదా సార్.
అవునమ్మా.
నేను రూపా వస్తాం సార్ కాలేజ్ దగ్గరికి.
షూర్ 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 రండి రండి.
సార్ అలాగే రికార్డ్స్ గురించి కూడా చెప్పండి సార్ ఏమైనా.
ఓకే, సో మీరు వస్తే నేను చెప్తాను మీకు ఎలా తీసుకోవాలి, ఏం చేయాల్సింది నేను చెప్తాను ఇక్కడికి వస్తే. ఓకేనా?
ఓకే సార్.
ఆర్ యూ కమింగ్ టుడే?
సార్ ఫ్రైడే వస్తాం.
Friday, you are coming Friday, on Friday. Okay, no problem.
Yes, sir.
No problem. Okay, ma.
ఓకే
So,
సార్ థ్యాంక్ యూ.
do you have any questions? ఓకే ఓకే. సో అబ్బాయిలు ఎవరు లేరా టౌన్లో నుంచి ఉన్న వాళ్ళు? ఓకే సరే, రూపా మీరు వస్తారు కదమ్మా, రూపా జ్యోతిర్మయ అండాల్. సో నో వరీ, మీరు రండి, మీరు వస్తే మేము చూపిస్తాం ఎలా ఉంటుందో, ఎక్కడికి వెళ్ళాలో చెప్తాము. యూ గో అండ్ గెట్ ది రికార్డ్స్. ఓకే, దెన్ యూ టాక్ టు యువర్ క్లాస్మేట్స్ అండ్ దెన్ ఫైనల్లీ యూ కెన్ ఫైనలైజ్ ఇట్. ఓకేనమ్మా?
ఓకే సార్.
ఓకేమ్మా. ఓకే, సీ యూ ఆన్ Friday then. Okay, have a good day.